Good morning, everyone. Today we are going to start with the new topic, which is the ECG. ECG, which is
Good morning, everyone.
known as the electrocardiogram. Electrocardiogram. Now, what is the electrocardiogram? Electrocardiogram, it is the technique. This is the technique by which the electric activity of heart is understood. So, someone called Pixel Reddy. Hi, good morning to you. So, this is the process by which we understand the electrical activity. So, in the last class, when we studied the conduction path, we did mention that pumping of the heart is the mechanical process, and every mechanical process needs the start from the electric impulse. So, that activity of the heart which is generating that impulse and it is manifested as a mechanical activity, study of that electric impulse is called as the ECG. Now, there needs to be a setup to study the ECG. To study ECG, the things required, because it is an electric activity study. So obviously, first thing that we will need are the electrodes, positive electrode and negative electrode, which will be connected to the galvanometer, the meter by whose deflection the electrical activity will be reflected. Third, we have is a pen which has like a sharp needle like point that will sense the electric activity and stress it. Fourth, we need a heat sensitive paper. Now this heat sensitive paper will sense the heat which is generated by the electric activity, and by moving up and down it will do the tracing. So through this activity, when we see the graph that is reflected on the ECG paper, we understand the electrical activity of the heart. Going by the diagram. So an ECG paper is something like this. So here we have the ECG paper. So clearly we can see that there are two distinct lines on the vertical line, that is the long line. Vertically we have this small square which is like 1 millimeter. So this paper is a 1 millimeter square grid. Now, each small box is like 1 millimeter vertically, that is like upwards, and horizontally it reflects the time. So horizontally, this one small square box, it is depicting time in second. And how much is it? It is about 0.04 second. So like that, if we consider one one big box, so how many square does it have? It is having 1, 2, 3, 4, 5. So horizontally, horizontally, one big square is 0.04 into 5, because there are 5 small boxes, equal to 0.20 second. This is the calibration. Now, on the top of each ECG paper, when we read the ECG, we will see there is a symbol like this. Now, this symbol designates the speed. This symbol designates the calibration. Now, this ECG paper, it runs at a particular speed. Speed at which the ECG paper goes on is 25 millimeter per second. That is the normal speed. Now, if the waves are small, then definitely the calibration will change. In that case, instead of 10 small boxes, here what we see is that 10 millimeter at a speed of 25 millimeter per second, the voltage that we get is 1 millivolt. Now, if the waves are small, then 20 millimeter will be equal to 1 millivolt. Reversely, if the waves are large, then automatically it will reduce. The normal is 10 millimeter. If the waves are large, again the speed will reduce to half. Then 5 millimeter will be considered as 1 millivolt. One very important point to remember is to make sure that all this setup, the electrical connectivity, the calibration, the speed, everything is maintained perfectly. Otherwise, the ECG waves that will be manifested on the paper cannot be taken as correct. That is very important factor. Now, coming to the ECG leads. ECG leads, there are 12 ECG leads. There are total 12 ECG leads. Out of that, 3 leads are bipolar limb leads. Okay, bipolar limb leads means these leads are connected through the electrode to both sides, like to two limbs. It can be the arm, it can also be one of the leg that is involved. Another 3 leads are unipolar leads. Like unipolar leads mean what? Only one electrode will be connected and another one will be an indifferent electrode. After that, what we have is the 6 precordial or chest leads. Now, the scientists who founded all these limbs, they were all been honored with Nobel Prize. This was like edge-breaking research in the field of medicine. Now, going by each and every lead one by one. To start with, we go with the bipolar limb lead. So bipolar limb lead, we know these are 3 in number. Looking at the picture of the bipolar limb lead. So here from the diagram, as you can see, there are like 3 limbs as mentioned earlier. So just have a look here. This is the lead 1, the first lead. This is the lead 2 and this is the lead 3. Okay, there are 3 lead. So if we look at the lead 1, it has got a positive input. It has also got a negative input. So if we write it like positive input, and here we write negative input. Now, one very easy way of remembering like in which lead, which limb is connected to the positive and negative. So here the whole ideology is that the heart is thought to be the center, the epicenter of the body, which is a volume conductor. This ECG was designed like keeping that in mind that the body is having body fluid and the body fluid has got cations and anions which act as volume conductor. The same thing that we mentioned in the start of our series in the general physiology part. Okay. Now, the target is to like study the electric activity of the heart. The closest region of studying the electric activity would be the shoulder area and the pubical area. But whatever activity, electrical activity that goes from the heart to the shoulder, the two shoulder will automatically be conducted to the limbs, and manually for a person to get connected with electrode at the limb is much more convenient than to get connected to at the shoulder level. Okay. Now, the 3 limbs that are involved, before we go into the leads, the 3 limbs that are used where that are used in the bipolar limb lead system to study the ECG. What are the things? They are the right arm, one limb, right arm. Then you have the other one, that is the left arm, and third, you have the left foot. Then what is this right foot used as? Right foot, it is used for earthing because electric impulse is going to get conducted. So this right foot is used for earthing. Okay. No electrodes are connected to the right foot. Now, a very easy way to remember which lead has the which limb is positive and which limb is negative. Instead of mugging up, always remember the right limb. The right limb, as you can see in the picture here. The right limb, that is the right arm. Here, in whatever lead we are using it, whether it is the first lead or the second lead. In right arm, it is only the negative electrode that is being connected. I repeat, in the right arm, it is only the negative electrode that is connected. Similarly, in the left leg only there is positive input, means only positive lead is connected. So if we remember that the left arm has only the negative input and the right arm has only the negative input and the left leg has only the positive input. So depending on the lead, the input to the left arm becomes positive or negative. Okay. Writing it down, how do we remember? Like the lead first or the lead 1. In lead 1, we have to remember it is only the upper limbs, like the right arm and the left arm that are involved. So, right arm, it always has what? It always has negative input. So, in case of lead 1, the right arm gets the negative input. See here, it gets the negative input. So, if the right arm is getting the negative input, obviously the left arm will get the positive input. Very simple. Coming to the next lead, that is lead 2. Lead 2, which are the limbs that are attached, that are linked, it is the right arm and the left foot. Again, it's very easy to remember. Right arm is always negative and left foot is always positive. Now coming to the lead 3. Lead 3, it is the input. The input is to the left arm and the right foot. So, and the left foot. Left arm and the left foot. So, the left foot, we know it is always positive. The left foot is always positive. So, obviously, the right arm is the one that is having the negative input. So, if we remember that the right arm is always having the negative input and the left foot is always having the positive input, and if we remember the limbs that are in the leads, we can automatically write on our own without actually having to mark it up. Okay. Now look at the connection. Now if this lead 1, lead 2 and lead 3, all these leads are actually joined. Like you see, these 3 are actually joined. What does it form? It forms a triangle. It forms a triangle. So this triangle, which has the heart in its epicenter, it is called as the Einthoven's triangle. Okay, it is called as the Einthoven's triangle. So this is our bipolar limb leads and how they are connected. Okay. Moving on to the next limb lead, which is the unipolar limb leads. This one is also called as augmented limb lead. So now by the terminology you can understand unipolar. It's unipolar. That means there is one input to like either positive or negative. Here in unipolar limb lead, there is one positive input to one limb, to one limb, and the rest two limbs that are not having the positive input, means no positive electrode is connected. Those two together makes a mean and forms the negative input. So it is something like this. So this is something, we drag it a little bit up. Yes, like this one. The augmented limb lead, as we can see here. Augmented limb lead. First, let's understand why it is called augmented. The terminology augmented means it is like powered up. It is like powered up. So here we can see the main lead that is receiving the positive electrode are AVR, AVL and AVF. Now here AVR means the R is like the right arm, right arm. Here AVR, the right arm has the positive input. and the mean of other two, means two are the limbs, that is the left arm and the left foot has negative input, makes the terminal for negative electrode. Okay, this is what it means. So AVR, by the term AVR, we have to remember the positive input is attached to the right arm. Second one is the AVL, is the AVL. Here the, as you can see here, the left arm has positive input and other two, just like how I described in the AVR, other two, that is the left foot and right arm has negative. Lastly, the AVL. The left foot has the positive input and the other two has negative input connected to the terminal. Okay. Now, if we look at both this limb, bipolar limb lead and this unipolar limb lead, we will see that this lead 1, it is taking the picture of the heart from the top, whereas the lead 2 and lead 3 are taking the picture from the bottom, from like the pubis area. Same is the condition in the augmented lead. Now, why it is called the augmented lead? It is augmented lead. The augmented leads have the 1.5 power as that of the limb leads, that is the bipolar limb leads. Another thing, point to be noted. I'm putting a star and writing. Point to be noted. Limb leads are on the they are on the frontal plane. They are all in the frontal plane, this limb leads. They are not in the horizontal plane. Okay. Next, the most advanced one and also very useful. All limb leads are very useful, but this is like very, very useful and pretty much in use, are the precordial or chest leads. First thing to remember, putting a star, these leads are on horizontal plane. Okay, this chest lead, these are on the horizontal plane. So how are they oriented? They are oriented like okay. If you look at the picture, you see this chest lead, they are nomenclature like chest leads are numbered from V1 to V6. They are numbered like from V1 to V6. And their orientation, how do they lie? They are like oriented in the intercostal spaces. They are placed in the intercostal spaces. So, looking at the picture, if we write the location of the leads, like V1 as seen here, V1 as seen here, what is the location of the V1? It is on the right side of the sternum. It is on the right side of the sternum on the fourth, lying on the fourth intercostal space. This is where it is lying. Okay, the first lead. Second one, V2. V2, if you see, it is lying on the same location like that of the V1. The only thing is that it is on the left side. It is on the left side. So, it lies on the left side of the sternum on the fourth on the fourth intercostal space. Okay, going to V3. So before writing V3, we have to understand V4. We have to understand V4. So here, looking at the V4, where is the V4? V4, it is, sorry, V4. It is placed on the mid clavicular line. So here you see, this is the clavicle. This is the clavicle. As you can see, this is your clavicle. So the mid clavicular line on the fifth intercostal space, on the fifth intercostal space, on the fifth intercostal space on the left. Okay. Now, after placing the V2 and V4, V3 is placed. Now, V3 is placed exactly midway between the V2 and V4. V4. V3 is placed like midway between the V2 and V4. It is placed midway between V2 and V4. This is where the V3 is. Again, before V5, we place the V6. So, going back to the picture, now V6, if you look at it, it is like the mid line from the axilla. It is the mid line from the axilla. It is placed along the axillary mid line on the left. Okay. After placing the V6, V5 is, after placing the V6, V5 is placed between V4 and V6. So, V5 is midway between V4 and V6. Now, all these leads that we have discussed take a different picture of the heart. We know the heart is lying in a slanting way. It's not like horizontal or vertical or linear anything. It is like lying on a slanting way, slightly towards more on the left side in the thorax. So all these leads, they are designed in such a way that the electrical activity from all sides of the hearts can be studied. So now, if we imagine that we have placed all the leads together, then what will be the ultimate picture that we get, ultimate image that we get from the leads of the heart? So this is a picture where all the leads are like placed together. So here you can see our limb leads 1, 2 and 3. These are the bipolar limb leads, right? We also have our unipolar, the augmented limb leads AVR, AVL and the AVF, the augmented unipolar limb leads. And then we have the precordial leads. We have the precordial leads here. All of them are not shown. It's like V1, V2 is not marked, but that is V2, V3. Then you have the V4, the arrow, V5 and then V6. Okay. So these are all the 12 leads that are placed together. Now, keeping all these leads together, if we have to understand which lead is actually showing the picture of the heart, or like taking the picture of the electrical activity of the heart from which area, it will stand out to be something like that. The lateral picture. Like taking the lateral picture of the heart is very important because it connects to a lot of cardiac problem, lot of heart diseases. We need to take the lateral picture actually. The leads that shows the lateral pictures are lead 1 from the limb lead, from the unipolar lead, augmented limb lead, again the left side. From the chest lead, we have the V5, V6. So these are the leads which is taking the lateral picture or lateral electrical activity functioning of the heart. If we study the ECG wave from these leads, it will give us understanding if there is any problem in the lateral side of the heart. Then the septal picture. The septal picture, it is best studied from the precordial lead V1 and V2. The septal picture is studied best from the chest lead V1 and V2. The anterior picture. The anterior picture is best reflected in the precordial lead V3 and V4. V3 and V4. The inferior picture. The inferior picture. Inferior picture means like somebody is taking the picture of the heart from the lower region, from the region of the pubis. The picture that we get is the inferior picture. So these are the lead 2, limb lead, bipolar limb lead 2, lead 3 and augmented lead F. Now coming to the, you must be wondering what happened to the AVR. AVR lead is, though it is useful in understanding certain waves, but in most of the cases, it is like a ritual thing. It is just like a ritualistic lead that is there with not much significance in understanding the electrical activity. Yes, in certain conditions, especially to understand T wave, we need that. We'll come to that later. But other than that, this AVR, this augmented chest lead R of this limb lead R, augmented unipolar limb lead R, that is on the right hand, it kind of takes picture from the top of the hollow area of the atrium. So AVR, this lead is not very helpful clinically. Okay, it is not very helpful clinically, and the findings from this augmented unipolar limb lead is always cross-checked with the other waves from the other limb leads and the precordial lead. So, this was our understanding of the ECG along with the various types of ECG, its implication and which leads actually give us which picture. Okay. Now, moving into the next part, which is the ECG wave. Okay, we have understood the lead. Now, what did we mention? What did we study in the before few minutes? That the electrical activity will be reflected on the ECG paper by deflection, by moving of the pen up and down, which will form the waves, which will form various waves, right? Now, what are the various type or the different type of waves? Now, ECG not only shows the waves, it also shows certain intervals and segments. So, before we go on to the picture, let's just mention the waves of ECG. Means what are the waves that is present, that is seen in the or present in the ECG? The waves are P, Q, R, S and T. It can also be written as caps P, Q, R, S, T. Okay. In most cases, we use the caps, but depending on condition, we use the lower case also, which again we will come after few things. Necessary to what does V? V is just like a nomenclature that is given to understand, to make us understand it is augmented, that it is like empowered. A is augmented, V is like given to designate the unipolar lead. This is done by the scientist. It doesn't have any otherwise meaning. What we have to remember is AVR means that the right arm has the positive input. Okay. Nishari. Okay. Okay. What? Ado Edin. Thank you for liking the lecture. So what is the meaning or implication of this P wave? P wave signifies depolarization of atrium, right? The heart has like two chambers, the atria and the ventricle, that you know. The atrium, the initial chamber, both it is signified by the P wave. Depolarization of both the atriums rather, let me write. Okay. What does the Q wave denote? Q wave denotes the depolarization. We'll come to the picture just after understanding that. Polarization of the ventricular endocardium. Like when from the AV node, the impulse is coming down across the interventricular septum, the first endocardial portion to get the wave of depolarization is the left endocardium. So it mainly signifies the depolarization of the endocardium first to the left and then to the right. So overall endocardium of the ventricle. R. R denotes depolarization of major parts of the ventricles. So major part of the ventricle, like the main mass of the ventricle, when it gets depolarized, as we studied in the last lecture on Saturday night. So that is what is the R wave. And S, it denotes the last depolarization of the lateral walls of the ventricles. Okay, the posterobasal lateral walls of the ventricles that swaps upwards, that is the depolarization reflected by the S wave. Now, what does T wave denotes? It denotes the repolarization of ventricle. And another known thing that we know, what is that? We also known that this QRS is a complex, right? Which is altogether denoting the depolarization of the ventricle. Okay, these are the waves of the ventricle. Now, if we study the picture and look into the and look into the other details of it, like as in this picture. Okay, so this is like the PQRST. We have studied, we know what is the P wave, we know what is this QRS complex, we know what is the T wave. Now here in the dotted line, we can also see one additionally another wave, which is like in dots, which is the U wave, Now what does this U wave mean? It denotes the repolarization of the papillary muscles. That is what it is denoting. Okay. Now, we have understood the waves. Now, apart from waves, there are certain segments and interval. There are certain segments and interval. Like the gap from the P till the R. Okay, not Q, R, because Q is so small, hardly it is visible by our naked eyes. So this gap, that straight line is the is the PR segment. Is the PR segment. How is it different from interval? We will come to that. Whereas starting of the P wave, all the way till the starting of the R wave, not Q wave. It is called as the PR interval. Okay. Now, right from here, let's have the concept of the segment and interval. So, this electric activity is nothing but the wave of depolarization which is traveling, which is being generated by the SA node, and then it is traveling through the conduction pathway that we studied in the last lecture. So, when it is only passing, like for example, from SA node to AV node, after contraction of the after contraction of the atrium, we get the P wave. But after the atrium has contracted, the time that it takes to reach via the AV node all the way till like the interventricular septum and then exciting the major part of the ventricle, the time gap, the travel of the impulse basically from the atria to the ventricle is not causing any contraction or relaxation, right? So that phase when the electric impulse is only traveling, not causing any excitation or relaxation, those are the straight line. Those are the straight line, meaning where only the electrical impulse is just traveling forward, okay, but not causing any excitation or relaxation, none of it. This is called the isoelectric line. So the segments are what? The segments are always the portion of the isoelectric line which is devoid of any wave. Means in a segment, in let me write it down here. In segments, there are no there are no waves, okay, but in intervals there is a wave and a segment, okay. This is the main difference. There is a wave and a segment. So as you can see here, going back to this picture, here in PR interval, we do have the P wave and the PR segment. Okay. Similarly, clinically, another important point that we'll be discussing very soon is the intersecting point where the S segment ends and another stretch of this straight line begins. Now, this segment where the S wave has ended and there is a straight, like as I have shown in the green, isoelectric line. The straight line is there. This area which is just after S segment and before the beginning of the T wave. After the S wave, before the beginning of the T wave, this area is called the ST segment. This area is called the ST segment. Okay. Then we have the next important interval or next important gap that we have is the TP interval. TP interval. So TP interval, this is of huge clinical implication. Now, this is after the T wave has gotten over and before the beginning of the next contraction of the atrium. So, literally, after the depolarization and repolarization of the atrium and ventricle one time and before the next contraction of the atrium, or in simple term, after one time the heart has completed its beating and before the next start of the beating of the heart. So that time, that duration is the TP interval. It has a U wave. So it has a U wave and the isoelectric line. Okay. Another interval that is also clinically relevant is the like the QT interval. This is the QT interval. Basically, after depolarization, the repolarization phase of the ventricle. So, basically, the repolarization phase of the ventricle, which includes the QRS complex. It includes the QRS complex because it is QT interval, QRS complex, the isoelectric line IST segment, and the T interval. Okay, this is one. Another referral point is the JT interval. Now, what is the J point? J point, as we mentioned here, the beginning of the ST segment, if we intersect, if we make a point that is like after the termination of the S wave and the beginning of the ST segment, this point is called as the J point. J point, very important as a referral point to study various clinical conditions like ischemic condition and disease of the hearts and other electrolyte imbalance. We will study in details. So this is the J point. Now, the gap between the J point and the T and the T wave, like the completion of the T wave. This is called as the JT interval. JT interval. So this QT and the JT interval, they are of clinical significance to study the ventricular repolarization. Okay. Since the R wave is very easy to see, good day to you also, Rishiraj. So, since the R wave is the peakiest one, it is the tallest one, and it is easily understood by our eyes. It can be easily read by our eyes in most of the leads. That also varies. We will come. So, here the gap between two R wave, the gap between two R wave is one cardiac cycle or one heartbeat. This is easy to calculate the heart rate from the gap between two R interval. Okay, so these are the important segment and the intervals. Now, we will understand each and every wave and this intervals and gaps in context to the clinics. So, to start with, we start with the P wave. We start with P wave. So, P wave is less than, what are like the characteristics of P wave? It is less than 0.12 millisecond. Like horizontally, it will occupy like just little bit more than like one small box. Okay, one small box, and P wave is less than 0.25 millivolt. Like it will occupy about like 2.5 small boxes vertically upward. Okay. Now, this is this P wave. P wave. P wave is best studied in the limb lead second and the precordial lead V1. So looking at the picture of it, yeah, no, okay. So these are the, as we mentioned, these are the picture of the limb lead 2 and V1. So actually, this is the first one. This is normal. This is normal P wave. So look at this picture. Understand this one. A normal P wave actually has like this bump. You can see this bifurcation here. The bifurcation here is just like two bump with a slight bifurcation. Why that is so? This is so that if you remember, what did we study? We studied like if we think this is our heart. Hold on. So this is our heart. Where is the SA node located? SA node is located in the junction between the superior vena cava and the right atrium. So it has to first generate the impulse, which first swaps the right atrium. From the right atrium through the bundle of Wenke bag, it goes to the left atrium, right? So it is the right atrium which is contracting little bit early than the left atrium. So this first bump, it is the depolarization of the right atrium, and this red one, it is the depolarization of the left atrium, because the electric impulse reaches the left atrium slightly later than the right atrium. In the precordial lead, that is the one that is taking the septal picture of the septum. So here also you can see it is biphasic. The blue one, it is designating the depolarization of the right atrium, and the one which is slightly negative, the red one, this is the depolarization of the left atrium. This is the septal picture, the picture from the V1. This is how a normal P wave looks like. Now, the second one, the second picture, this is our picture 1. Now, this one is our picture 2. Okay. Now, this picture, this one, this is actually a picture of P mitrale. Like, how does the wave look in case of P mitrale? Now, what is P mitrale? Here, stenosis of mitral valve causes enlargement of the left atrium. So mitral valve, we know it is present on the left side of the heart. So it is causing stenosis, blockage, the narrowing down. Usually it's a congenital disorder. It's narrowing down causes the enlargement of the left atrium. So when it causes enlargement of the left atrium, the mass of the atrium on the left side tends to become more. So here you see the same bifurcated V wave becomes distorted. The hump is more on the left atrium, that is designated on the picture on our right side, right? So here the depolarization of right atrium and Here it is the depolarization of the left atrium. It becomes more enlarged. It becomes like a more hump. And on the precordial lead, the size of the depolarization. See, this dotted line means this is the normal one. What it should be, this dotted lines, if you see this black one, this is how it should be. Instead of that, there has been a larger hump. There has been a larger hump of the left atrium wave in case of V mitrale. Okay. Similarly, in the V1, you see the amplitude is the has become like slightly steeper, and here the biphasic is like little bit lagging below. It should ideally been it should ideally been like this dotted lines, but here you see the depolarization of the left atrium is like little bit more biphasic going below. So this is the depolarization of right atrium, whereas this is the depolarization of the left atrium, which becomes more negative. The third situation, the third condition is like P pulmonale, the third one, the third one. So what is this P pulmonale? Please remember here, this one, here, this one. So P pulmonale, it means there is stenosis of the pulmonary valve causing right atrium enlargement. Okay. So here you see the biphasic structure. Here the one which should be biphasic, this is gone. Instead there is a big and there is a much more prominent and big hump. Only one single P wave is visible. So which signifies the depolarization of right and left atrium, that is on the lead 2 and on the lead 6 also. The summing up of this P wave happens, and it is not at all biphasic. So here you see the hump is like taller, the P wave becomes taller. It should be like this, but it becomes taller, and there is a single P wave, not double. So this is the depolarization of the right and the left atrium on the lead 6. Okay. Now, this is all like talks about the depolarization. Nowhere in the ECG graph did we mention or see or notice the repolarization of the atria. So, why we don't see the repolarization of atria? Why no repolarization wave of atria? Because the repolarization T of atria is very small, and because the mass of the atria is much smaller than that of the ventricle, right? Therefore, it is buried under the QRS complex. So it is not at all visible. Then then when is the atrial T wave visible? Just simply for now, what we have to remember, when is this T wave of atrial or atrium is visible? In condition, of course, in clinical condition, in pathological condition like acute pericarditis, okay, atrial tumor, or conditions like atrial infarctions. So in this clinical condition, we will get the T wave, that is a repolarization of the atrium, which is, if there is a depolarization wave of the atria, it is always abnormal and it will always be due to some clinical condition or the other. Also, a low amplitude T wave is seen in AV log. Whenever there is delay in the conduction of impulse, there is this appearance of this low amplitude atrial repolarization. Okay. So now we have a good knowledge of the P wave. Now, what follows after the P wave? What follows after the P wave is the PR interval. PR interval. Now, this PR interval is about 0.12 to 0.22 seconds. That is how it should be on the ECG. 0.12 to 0.22 seconds. So here, if the PR interval is greater than 0.22 second or 0.2 means 0.20 second, then it will indicate a lock or hindrance in the conduction of impulse. Similarly, if the PR interval is lesser than 0.12 second, then it will mean presence of an extra bundle that exists in the bundle or extra pathway in the conducting region. Okay. This let us see and understand with this graph, with this diagram. Okay. Now, as we can see here, the first diagram, our first diagram, that is the normal AV impulse transmission. This is what we studied in the last class. Like here, this is our SA node. This is our SA node. Okay. These are the internodal bundles, and this is our AV node. Okay. Now, this is the picture, the leftmost side. This is a picture that is showing a normal transmission or conduction of impulse. Impulse is first being generated from the SA node, and as we see through the arrows, via the three internodal bundles, it comes to the AV node and also goes to the left atrium. This is right atrium, left atrium, and this is the right ventricle and left ventricle, and it follows a normal pathway that we studied, like through the bundle of his bundle of Purkinje fibers, the fascicle. It is swapping the or transmitting the depolarization wave to the ventricles. And here we see the normal PR gap, the normal PR interval is of 0.12 to 0.22 seconds. This is what is the normal scenario. Now, if a person is having first degree of AV block, now this first degree of AV block, it usually happens if all of a sudden there is withdrawal of the sympathetic tone, or if the vagus nerve becomes more active, as seen in athletes. Athletes, because they are practicing, they are undergoing physical exercise daily in a rigorous way, the vagus nerve becomes very active, their vagal tone increases so that the heart rate is under control. So, on one such event, if the vagus nerve inhibits the SA node, the vagus nerve, it also innervates the SA node, right? It innervates the SA node and the AV node we studied in our last previous class. So, if the vagus nerve starts controlling the SA node more, then the conduction the conduction of the impulse will take longer time. This is one reason. Or if there is like some kind of blockage in the internodal pathway from the SA node to the AV node, if there is like some kind of blockage in the internodal pathway, as we have seen, or the vagus nerve has like done its function or stimulated, like has stimulated its secretion, or rather if there is vehicle inhibition on SA node, then what will happen? Then also the impulse to come from the SA node to the AV node will take a very long time, or there can be some blocks also on the pathway. Maybe there are some blocks in the pathway of transmission. All this thing can lead to longer time for the impulse to travel from the SA node to AV node. In this condition, what happens? The PR interval becomes more than 0.22 seconds. Okay. There can be a third scenario, means the pre-excitation. Now, pre-excitation, again, we have mentioned about it in the last class. What is pre-excitation? Say, due to some reason, there has been a generation of a extra conducting bunch, a accessory pathway which is capable of conducting the impulse, which is termed as the which is termed as the bundle of Kent. Like if such extra accessory pathway is generated, then what will happen? What is the scenario we studied? Like from the AV node, as usual, there will be transmission of impulse. There will be transmission of impulse, right? But as soon as this impulse that it reaches this bundle of Kent, it will, instead of going into repolarization, this one will again pass through this conducting region and again enter the ventricle. So there will be excess, there will be an extra impulse that is entering the ventricle even before the SA node has generated an impulse. I repeat, if there is presence of an extra accessory pathway like bundle of Kent. Now this bundle of Kent is capable of transmitting or conducting impulse. So what happens? When the SA node has generated the impulse, first it will follow the normal course. It will go via the interventricular septum, and then the wave of depolarization will complete its course and go all the way posterior basal laterally upward, right? After that, When it goes, say here on the right side, right lateral side, there is bundle of Kent. So when it goes to the right lateral side, it will find the bundle of Kent. So instead of ensuring repolarization, this current will again re-enter the ventricle and will excite the ventricle abnormally. So here the impulse is taking much shorter time. Due to the re-entry of the current. Now here this PR interval is less than 0.12 second, and one characteristic of this pre-excitation is the presence of this delta wave in the ECG. So whenever there is any pre-excitation, there will be drastic change in the time gap of this PR interval. It will be lesser than 0.12 second. Additionally, on the ECG, we will see like a buried, some wave is buried in the R wave, which is the delta wave. Okay. This is the clinical implication of the PR interval. Now, after the PR interval, the wave that comes is the Q wave. Now, the characteristics of this Q wave is that Q waves are usually not seen in V1 to V3 precordial leads. It is or may be visible at the AVR and the inferior lead 3. Here, the size is less than size of what? Size of the Q wave. Less than 2 millimeter in amplitude. Okay. Whereas in V5, V6, septal Q wave may be present. Now, in pathological conditions, the Q wave can be as big as 40 millimeter. It can be very prominent. It can be as big as 40 millimeter. Absence of Q wave signifies absence of Q wave signifies where? Absence of Q wave in the V5 and V6 signifies left bundle branch block. Okay. So, a few pictures over here. Wait. No, okay. So from this picture, we will understand. So in most of the cases, as you can see in the lead 1, 2, 3, the limb leads, and then we have the precordial leads. So here distinctively you see in V1, V2, V3, we cannot see a Q wave that is visible. Okay, whereas in V5 and V6, we do find a very small and tiny, tiny septal Q wave. Just before the R wave, if you see, you can find tiny, very tiny, yet little bit visible septal Q wave. Okay. Only in pathological condition, the Q wave becomes deeper and prominent. Next wave is our R wave, which means the major portion of the ventricle has depolarized. So the first negative wave was the Q wave, and the second positive deflection, as we have seen, that is after the P wave, is the R wave. Now typically R wave increases in size, that is in amplitude, from V1 to V6. Like the amplitude is smallest in the V1 and then it increases to V6. Here, R wave is, how does ADO18, how does atrial polarization appear on ECG? As I told you, 18, atrial repolarization, I think you mean, right? Atrial repolarization is not seen in normal condition. Only if there are abnormal condition like tumor infarction, tumor of the atrium, or an infarction of the atrium, only then you see the repolarization. And how the depolarization appears, of course depolarization it appears because it is the contraction is a much more forceful thing, and it is the beginning of the contraction, beginning of the systole. So the electrical activity is much more enhanced. If you see, all the depolarization waves are much bigger compared to repolarization wave, right? Look here, even for the ventricle, the re depolarization is always much more bigger than the repolarization. Repolarization is only T. So atrium also, the depolarization is evident. The P wave is evident, but the repolarization, it happens at the almost the same time when the depolarization is happening. So it is like buried under the QRS complex. That's why you don't see the repolarization of the atrium, but you see the depolarization of the atrium, because when atrium is depolarizing, ventricles are quiet. It's not undergoing anything. Means it is undergoing diastole. It is not undergoing systole. Understood? It has completed the diastole. Only after a gap of certain time, then the depolarization of the atrium happens. Hope you understood, Aideen. So coming to the R wave. R wave increases from size from V1 to V6, and R wave is visible in all leads. It is visible in all leads. Okay. Now the max peak time for R wave is like between, taking both the right ventricle and the left ventricle, it is between 0.035, between 0.35 to 0.045 second. This is the time where the R wave is like maximum in normal individual. That is the peak of the R wave. Going by the amplitude, the size, the amplitude of R wave is less than 35 millimeter. Okay, less than 35 millimeter. It maintains, it is about 26 millimeter in healthy individual. That is usually the value of it. Now, R wave, it signifies the maximum area of the depolarization. Now, clinically, the R wave in V6 and V5, V5 and V6, and the steepest or the tallest S wave in V1 is compared and used for diagnosis. Like in most of the normal condition, if you sum the amplitude of the S wave in V1 and amplitude of the R wave in V5 or V6, it should be never more than 35 mm, that is vertically. If it is less than 35 mm, then it means the cardiac function, the electrical activity are normal. Why electrical activity normal means cardiac function is normal? Because if electrical activity is normal, that means the contraction and relaxation of the cardiac muscle is normal. So if the contraction and relaxation of cardiac muscle is normal, that will mean there is no clinical condition, there is no blockage, there is no condition like atherosclerosis or ischemic condition or infarction or any kind of tumor, any kind of interference in the functioning of the heart. Okay. Next, we will study the S wave and then we will use the diagram to understand. Next is the S wave. What is the S wave? It is the second negative deflection. It is the second negative deflection on the ECG. So, this S wave is like about or less than like 0.2 to 0.4 millimeter. Okay, it is very, very like small. It can be the max up to 2 millimeter. Maximum it can be up to 2 millimeter. What does it signify? It is the depolarization of the lateral walls of the ventricle. Okay. Now, S wave, why is it particularly important? This S wave is particularly important clinically in context to in context to J point and ST segment. We'll study all lot of it just after 1 or 2 minutes. Okay. Now starting the QRS complex. Okay. So what is the QRS complex? This QRS complex, it is like studying the entire depolarization of the ventricle together. This depolarization, this QRS complex the depolarization of the ventricle is about 0.06 to 0.10 seconds on the time scale, that is on the horizontal scale, that is the width of it. Okay. And if we specifically measure the width of QRS is between 70 to 110 millisecond. Now, if the QRS is narrow, is less than 70 millisecond, it may indicate supraventricular tachycardia. Okay, maybe some ectopic foci has generated and it is causing extra beat. Okay, so supraventricular tachycardia. If the QRS is greater than 110 millisecond, it may indicate ventricular hypertrophy. Okay, it may indicate ventricular hypertrophy. The ventricle would have become thick, the walls of the ventricle would have become thick and enlarged due to some reason. Electric alterance means instability in the electric recording, like in the voltage and all, in QRS may be seen in pericardial effusion. So, pericardial effusion means in the interpericardial region, there is accumulation of the pericardial fluid. So, this accumulation of the pericardial fluid will cause the heart to swing back and forth, back and forth. In that condition, the base, like the base that is usually, our ECG base, right? This base will become wobbly. The base will become like wobbly. The graphs will also, it will become like kind of not having the same kind of base, and it may go up and down. So there will be that wobbly kind of this ECG waves whenever there is any change in the voltage due to the pericardial effusion. This is the main characteristics. Now, reading a QRS complex is very essential and one has to know how to read a QRS complex. Now, depending on the size, the QRS are either written in uppercase, okay, means in capital, or in lower case, means in small hand. Okay. If there is only negative deflection, if there is only negative QRS, then it is QS complex. Then it is called QS complex. Another characteristics of the QRS is that if two R waves are seen, then the second R wave is written as R complement. Now, few of this example we will study with the diagram. So, see here. Okay. So this is a chart, like this is like an amalgamation of the diagrams that is showing the different type of QRS and what do we call it and why do we call it. So here you see in the first one, there is only one positive deflection. After the P wave, there is only one positive deflection. So if we see one positive deflection and the amplitude, the size of the wave is bigger, then we call it as capital R. We call it as capital R. Why capital R? Because this positive wave, it is bigger in amplitude. That's why positive R. Next case, here you see, we can see two waves. One is the upward stroke, which is positive, and second one is the downward stroke, which is below the isoelectric line, negative. So obviously the first upward stroke is the R wave, as written here, R wave, and the amplitude of the R is bigger, so capital R. So capital R. Whereas you see the S is having a small amplitude. It is lower case s. So here we read it as RS. For example, here this is like the R is smaller, just like how we see in the initial precodes, like in the septal precordial leads, or in most like V1, V2 type of the leads, we see such type of a R. With the R, it's a positive deflection, but you see it is small in amplitude. Whereas it is followed by a very big and deep S. So here the R is small, the R is in lower case, and the S is capital in the upper case. Next is a typical QRS complex, in which the R is the loudest, the R is like the picky one, the top is the most prominent one, and the Q is a small deflection, so is the S. So here only the R is capital, whereas the Q and the S both are in lower case. Moving to the next wave, here you see there are two, like one positive and one is negative. So the first negative deflection is always Q, and the first positive deflection after the P wave, that is the second positive deflection on the ECG after the P wave, it is called as the R. Okay, so both this Q and R, they are very big in amplitude and they are occupying many small boxes. So this is called as the Q and R wave. See the criteria of someone called Tom Cruise. It would be better if you write your real name. The main criteria to call capital uppercase and lowercase is just as I am explaining, to see how big, what is the amplitude of it compared to the other waves, compared to the Q or the S that is present. Okay, the size by your eyes you can see and you can understand whether the R is bigger or smaller than the S wave or the Q wave. Next comes the QS complex, as we mentioned earlier. If the QRS is only having a negative wave, after the P wave, if there is no other positive wave, but it is only a negative wave which is big, and there is no positive wave within it until the T wave is there, then it is called as the QS complex. Okay. The third one, as you can see, after the P wave, first there is a large and big down deflection, the negative wave, which is the big Q wave, followed by a smaller R wave, which is a smaller in amplitude. By your eyes, by the size you can see, Tom Cruise, whatever your name is. So the smaller R wave, which is smaller in amplitude compared to the Q wave. So here we write the R as small and the Q as big. Okay. Next, we have the concept that I told you earlier. Like if we have two R wave, we call it, the first one we call the normal R and second one we called as the complement. So see here, first understand, see, first look, first there is a positive deflection obviously after the P wave. Okay, let me use a smaller pen, thinner pen. So after the P wave, you see there is a small positive deflection, that is obviously the R wave. Now after the R wave, you see a negative deflection that has got below the isoelectric line. That has gone, this is your isoelectric line. So it has gone below the isoelectric line. Okay. So this is obviously S wave. Only the S wave goes below the isoelectric line. So R S, and then you have a bigger positive deflection for the second time. So such bigger positive deflection is of the R wave only. That means the ventricle is again depolarizing. First there was a smaller depolarization, followed by the lateral wall depolarization, and something, maybe re-entry of a current, has caused the again depolarization of the major area of the ventricle. So the first one is smaller compared to the second R wave. So the first R wave we write as small r, and second one as bigger R, and second R we have a complement. If there are two R, to signify which R has been visible later, has come to the ECG gram later, we write it as complement. So small r, smallest, and capital R complement. Okay. Next, we have a Q and R wave. A small Q, a small negative deflection, followed by a sharp and big R deflection, positive deflection, Q and R. And this is a picture of a notched or bifurcated R wave, like a broken. There is no S, there is no Q, nothing. So here you see this R wave, only there is one R wave, but that is also not sharp and complete. It is like notched. Okay, it is notched. Then next you have is R complement. This is also again a point to understand. See here, the first we have is a R wave which is positive. Now this graph, this R wave, the end of the R wave and the beginning of the other R wave, this point, if you see, it has not gone below the isoelectric line, right? It has not touched the isoelectric line and gone below, like this one. Here you see the S wave, it has gone below the isoelectric line. But here this wave has not gone below the isoelectric line, so we cannot call it a S wave. We cannot call it a S wave until and unless the negative deflection has touched the isoelectric line and gone down. So here there are only two R. The second one is complementary. The second one is bigger in size, so it is small r and R complement. Okay. Third, what we have here is, let me write it out. What we have here is fragmented R waves. Okay, so these are the various criteria, and these are the various types of the QRS complex. Okay, hope you understood. In case anyone has a doubt, can type out to me. From Chris, I hope you understood. Yes, in the bundle bunch, in the left bundle bunch, there will be no septal Q wave, right? And also in the right bundle bunch, the R will not be of the same amplitude that it is there. Okay, the whole QRS complex will not be of the regular type, and the Q wave will be missing in case of left bundle bunch. In case of a right bundle bunch, we will come. It will not only affect the QRS segment. The main thing to understand the left and the right bundle bunch is usually the ST segment, which we will study now. We have pictures of the ECG to understand that. Okay. So after the After this QRS complex, we move on to, we have understood QRS. Now comes the important and very interesting part, which is the understanding the ST segment. ST segment. Now, what is the ST segment? ST segment, it is in context to the J point, the segment Segment means the isoelectric line between the S wave termination and T wave beginning. That means after the S wave has ended and the T wave is about to begin. That isoelectric line, it is the ST segment, and it is in context to the J point, the point where the S wave ends and the ST segment begins. It is called as the J point. Now, this J point or the ST segment, this ST segment is referral point in understanding ST segment depression and elevation, like whether the ST segment is like higher than the isoelectric line or it has gone below the isoelectric line. But here also one thing to be remembered is, if the depression or elevation is about 0.5 mm, like half a small box from the isoelectric line, that half a small box, if it has gone up or down, then it is not clinically relevant. Means it does not indicate any clinical condition. We cannot say that the person is suffering from some clinical condition if it goes only half a box up or down. And always remember, ECG is not only like the final diagnostic pinpoint kind of thing. It has to be correlated with lab study and imaging, and then only a proper diagnosis can be made. But it gives a very good understanding of the possibilities that a person may be suffering from. Okay. Now, usually conditions like ischemia, myocardial infarction. Of course, the symptoms also has to be correlated. Hypo and hypertrophy Kalemia. Hypo and hyperkalemia. Supraventricular tachycardia shows ST segment deviation. Okay. Also, hyper, these are the conditions before we go and study the ECG patterns. Ventilation, exercise, and pregnancy. These are the non-clinical conditions can show ST segment deviations. Okay, now let's study some of the examples. Now, so first we start with the ST segment elevation. Okay, before that, I want to show you something else also. Okay, so show you something else first and then go to that one. Okay. So here, if we see this just for our better understanding, like how does we understand this ST segment elevation and depression? This is totally in context to the J point. As I told you, this is totally in context to the J point. So this is the J point, which is actually the intersegment between the S point as well as the S termination of the S wave and from where the ST segment has begun. So now here you see this is the isoelectric line. So this is actually the baseline, the isoelectric line. So here the J point is elevated. See here J point is elevated as enlarged and shown very clearly. It is elevated by like 4 boxes. It is elevated from like isoelectric line by 4 boxes, like 4 boxes. Now 4 small boxes equal to 4 mm of ST segment elevation, which is more than 0.5 mm of elevation. So this is a clinical condition. Another one, as seen in case of ST segment depression. Again, see, this is the isoelectric line, right? It should be here. Isoelectric line, it should be here. So here you see the ST segment. This is the J point. This is the J point. Here, the ST segment, it has gone down the isoelectric line by 3 millimeter, like 3 small boxes. So it is a ST segment depression by 3 millimeter. Okay, this is like the elevation and the depression, and few examples we are going to study. Now, one thing usually point to remember, let me just write it out. Point to remember is that usually ST segment elevation and depression is followed by an abnormal T wave. So understanding the T wave is also very important. A few examples that we will study here are, okay, let's start with the ST segment depression. No. So here, these are the few examples of the ST segment depression. Okay. So as you can see here, compared to this is our isoelectric line, as I have told you. So compared to this isoelectric line, the ST segment has gone like below it. See, this ST segment in all these cases, the ST segment has gone below the isoelectric line. The various pattern of it, like the ST segment after going below the isoelectric line, this one is horizontal, this one has a down sloping, giving a shape of a downsloping. This one is linear, this one is again, you see this one is shape of a concave. Okay, this is also downsloping concave, this is also like downsloping kind of a concave. These are all the typical example. These are examples of ischemic ST segment depression. Now here, if you notice one more thing, here in this condition, there is also the T wave. There is also this T wave inversion. T wave inversion. Now coming to the horizontal type. Here in pregnancy, hyperventilation and heavy exercise, this can also be seen. This type of horizontal, like depression that has gone horizontal, it can also be seen in physiological condition like pregnancy. If a person is breathing heavy or if a person is exercising, then also it can be seen. Now, the downsloping type with a very prominent T wave. Here you see the T wave is prominent. Okay. This can also be seen in patients who are suffering from hypokalemia. And these are the examples of a typical ischemic heart condition. Now, here, this is the example, this one, it is an example of D. Winter's sign. So, there was a time when it was thought that all the ischemic condition Tom Cruise, if J point is isoelectric, no, it will never happen like that. There will not be a condition that the J point will be isoelectric and the ST segment will go up and down. If the ST segment is going up and down, automatic the J point will go up and down, because the beginning of the ST segment itself, see here, the beginning of the ST segment itself is the J point. So it will never happen that the J point is correct and the ST segment has gone up and down. Okay, it never happens like that. So here in this D Winter sign, what happens? It is the first of its type. Earlier it was thought that these ischemic conditions are always like downsloping. But in a certain population in the Europe, it was seen that they are having a ischemic heart condition due to disease of the coronary artery. Yet this ST segment is depressed, but it is of upsloping type. It is of upsloping type with a prominent T wave. Here it is upsloping ST segment with prominent T wave. Okay, this was the first type that was found out. Now here below we see example of various condition of the blocks. So this one is in lead V6, which is the left bundle branch block as studied. Here you see the fragmented R wave. The ST segment is like downsloping depressed with a inverted T wave. Now here you see V6 left ventricle hypertrophy. Okay. Here this picture is of V1 right bundle bunch block, where you see there is a much prominent Q wave. Prominent Q wave here in the left bundle block in the V6. What did I tell? The Q wave will be missing. So here see no Q wave. But in case of the right bundle bunch block, you see the Q wave is much prominent with a small R wave and a downsloping ST segment and inverted T wave. Okay. This one should remind you of the delta wave. So this is an example of the pre-excitation, and this is a typical picture of the V1 lead showing the right ventricular hypertrophy. These are few examples of the ST segment depression. Now, same way, we will study some example of the ST segment elevation. So, this ST segment elevation can also be used to study various ischemic condition, non-ischemic condition and condition of the heart. So, this is ST segment elevation. This is ST segment elevation. Again, if you see this is the isoelectric line. Our isoelectric line, let me correct the pen. Isoelectric line. See here, the J point is elevated in all the picture. I have marked in some, rest you can understand. It is elevated, and there are various pattern of the ST segment. Some ST segments, it is of the convex type, as mentioned here. It is of the convex type. This is straight up sloping. Here you see this is horizontal, elevated yet horizontal, and this is down sloping. These are the typical example. These are the typical example of ischemic heart. Okay. Here another, this is a non-typical ischemic heart. So here you see this ST segment is obviously elevated. ST segment is obviously elevated, but it is like of the concave type. The ST segment is concave type. Now here down you can see these are like more examples of ST segment elevations. So here in some you see it is elevated with a very big T wave, that is also elevated. Here you see again concave. The next you see which is like horizontal, and this is like T wave inverted, right? Here again you see something like the D winters where a loud T wave is present and it is like upsloping. The ST segment is upsloping. Here again you find upsloping with a concave T wave, and here you find a upright, up straight T wave, which is followed by a ST segment, which is horizontal. These are the various examples of the ST segment. Now, on your right side of the screen, let me partition it out little bit. It will be easy for you to remember. So, this is a typical example of a person who came to the clinics with chest pain complaint. It is actually a case of myocardial infarction. So this is a typical case of a person who was a driver by profession and who was about like above 50 years of age. So when he complained of the chest, ECG was done immediately and later on it was confirmed of myocardial infarction of the ischemic type. So what was seen? It was seen that the two opposite limb, right, like the reciprocal limb, like AVL is reciprocal to the limb lead 2. Here in one limb there is ST segment. Here there is ST segment depression followed by elevation. Same thing. In the reciprocal limb, in one there is depression, in the other there is elevation. Same in all the lid, comparison was made. One, there was elevation. Second, there was like, depression. So, whenever there is a myocardial infarction and immediately ECG is done, it is seen that in one lead there is elevation and in the reciprocal lead there is depression. This is how a person who suffers from anginal pain comes and does the ECG immediately, and the ECG reflects the waves of this pattern. Okay, this is our ST segment. So remaining is our T wave and the U wave. Coming to the T wave. T wave is also very clinically important. Okay. Now, essential characteristics of T wave is that T wave is upright in all leads other than V1 and AVR. These are the two lead in which the T wave is not upright. It is inverted. So straight T wave in V1 signifies coronary disease, some problem or the other. T wave more than 25% in amplitude of the R wave signifies hypertrophy of the ventricles. Okay. Now, this T wave is affected in the hypokalemia, in myocardial infarction, in ischemia. We already saw like anything or condition affects the ST segment can affect T wave. Like T wave can be inverted, T wave can be concave, it can be too loud. All this thing we have already seen. A few examples of this condition of this T wave. So this is a this is a hyperacute T wave seen in ventricular hypertrophy. So if there is ventricular hypertrophy, it may cause to such kind of a peak. Like this is the T wave. This is the T wave. This is the T wave as shown in the V3. So you see, it is almost as tall as the R wave. It is like very big and hyper acute. So this kind of hyper acute T wave will be seen only when there is any hypertrophy of the ventricle. More clinical conditions affecting the T wave, as we see here, is like this one. We have studied various examples of the T wave which has gone abnormal in the ST segment depression and elevation ischemic condition. Now, this is the here. This is an example of Prince metal angina. Now, what is Prince metal angina? Here, ischemia is caused due to spasm of the coronary artery. So, if there is like spasm of the coronary artery, then there is this ischemic condition which causes a very loud and prominent T wave. This T wave is like broad and peaked and wide. It is like very loud as seen in the V2 and V3. Since it is V2 and V3, you see the R wave is hardly visible. It is kind of like masked due to the T wave. So here you see in Prince mental angina also you find a very loud and prominent T wave. Again, more examples of this abnormal T wave. One more we are going to study. That is that as seen in case of Valence syndrome. Here, biphasic T wave is seen. So you can see here the T wave has gone both up and down the isoelectric line. Up and down the isoelectric line. This is biphasic T wave. And in case of electrolytic imbalance also, in case of electrolytic imbalance like severe hypokalemia. Okay. Example, severe hypokalemia. This camel hump T wave is seen. Okay. These are the few abnormal T waves that you see in various clinical conditions. U wave is also equally important. So, U wave, what is its characteristics? usually 0.2 to 0.4 millimeter in size. Okay, it is prominent in V2 and V3. Prominent in conditions like U wave usually in normal condition is not visible, but it becomes more prominent in condition like bradycardia. When the heart rate slows down, the U wave becomes very prominent at 65 beats per minute. It becomes very prominent. Then in hypokalemia also it becomes very prominent. It also becomes prominent in increased intracranial pressure, in myocardial infarction, ventricular hypertrophy. It also becomes prominent in pulmonary embolism. In all this condition, the U wave becomes very prominent. One example of this U wave is this. So, this is a condition where you can see a prominent U wave. Example, hypokalemia. Now, here the U wave is preceded by a large and prominent T wave. Okay. Lastly, our another important segment or interval is the QT interval and JT interval. So, both these interval signifies the repolarization of the ventricles. Okay, it signifies the repolarization of the ventricles. Clinically, the JT interval is more used. Third, these interval like both QT and JT intervals, they increase when the heart rate decreases, that is during bradycardia, and they decrease during tachycardia. So during tachycardia, they decrease, and during bradycardia, they increase. So what is the diagram, or what is the location of this JT and the QT, just for a quick remembrance? So this is our graph. The QT is the start of the QRS complex. So QRS wave, QRS complex, all the way till the completion of the T wave. This is QT interval. And JT, this is J is the referral point. The J point is the referral point. And JT, it is the time from the start of the J point till the till the T wave gets completed. So this is the JT interval, and both are important because they signify the repolarization of the ventricular muscle. So QT signifies, QT is like depolarization plus repolarization, though it is used mainly to study the repolarization. That is the reason why I wrote it is mainly to study the repolarization. Okay, and JT interval, it is the repolarization of the ventricle. So clinically, JT interval is more significant than QT interval in studying the repolarization of the ventricle. And why repolarization of ventricle is essential? One point to remember: repolarization of ventricles are essential for proper filling of the ventricle. So if ventricles don't fill up with adequate amount of blood, then there will not be enough blood that is pumped to the periphery and the pulmonary circulation. So our class of ECG, I hope we have laid a very good concept. We have understood what is ECG, what is the setup in brief, and then we gone into understanding the leads and how they are set up, on which planes. We then studied the orientation of the leads, their nomenclature, their connectivity. Then we moved on to the ECG, the weights, the segment, the interval, and their significance with their clinical implications and correlation. Next class, we would study the cardiac cycle. Okay. So till then, bye and thank you all.